U.S. Agriculture Secretary met Governor Abbott today to announce steps to battle an invasive species that could threaten Texas livestock. That invasive pest has been spreading north through Mexico.
People in Nevada were shocked when millions of bees were released into the desert. At first, many laughed, not knowing what was coming. Some even said the plan would fail within weeks. But as time passed, the story took a surprising turn, leaving people silent and experts racing to explain what happened. But the real twist wasn't just what happened in Nevada. It began decades earlier with a bizarre plan involving tiny flies and fire ants. Why would scientists even try this? How the idea started. Red, imported fire ants carry the scientific name Solenopsis invicta. They arrived in the United States in the 20th century and spread through the southeastern states. Farmers, utility workers, and homeowners all saw the same problem. Young poultry and small livestock sometimes died after severe ant attacks. Sometimes ants were found inside electrical equipment, which caused malfunctions and outages. Parks, sports fields, and schoolyards are filled with round ant mounds. Chemical baits and sprays helped where they were applied, but they were expensive and hard to use across whole counties and regions. That problem pushed researchers to look for a different option. During the 1980s and 1990s, a team of entomologists planned a new approach. Scientists from the United States Department of Agriculture Agricultural Research Service, the University of Texas at Austin Brackenridge Field Laboratory, and Texas A&M University joined with entomologists in Argentina and Brazil. They traveled to the ants' native range to search for natural enemies. Field collectors found several tiny flies that attack fire ant workers. The flies belong to the genus Pseudactyon. Specialists showed how the flies moved and hunted along ant trails. Field observations in Argentina and Brazil gave a clear picture of how the flies acted. Adult flies hovered near foraging trails and often struck a worker. The attacked worker left the line of ants and moved into the shade or into a crack in the soil. The fly laid an egg or deposited a larva that developed inside the ant. Over days, that larva killed the host worker. Researchers also saw a second effect in the field. Where fly populations were common, fewer workers left the nest to forage during the day. That change suggested the flies did more than kill a few workers. They also altered ant behavior in a way that could reduce food gathering. Turning the natural history into a control method required careful testing. United States laboratories brought candidate Pseudactyon species into quarantine and exposed them to a range of North American ant species. The purpose of those trials was to show that the flies preferred Solenopsis invicta and would not attack native ants. They recorded which ant species the flies approached and whether they attempted to lay eggs. Only fly species that showed a strong preference for the invasive fire ant moved forward in the program. The testing aimed to avoid harm to native insects. Regulatory review followed the tests. State agricultural departments and federal agencies required formal release plans. Researchers prepared detailed protocols that described how they would rear flies, how they would transport parasitized ants or adults, and how they would release them on field sites. Permits set limits on transport and required steps to prevent accidental escape during handling. Extension agents and university outreach staff briefed landowners and officials so everyone at release sites knew what to expect. Technicians then built rearing programs in secure facilities. Laboratories kept fire ant colonies healthy and fed so flies could continue their life cycle. Staff learned to handle parasitized workers without harming the developing fly. Release methods included placing small cages of parasitized ants on active mounds and releasing lab-reared adults along known foraging paths. Field crews logged every release with the date, the species name, the number released, and the exact location. They recorded weather and habitat features because temperature and humidity affect fly activity. Field releases began in the late 1990s. The first reported establishment of Pseudactyon tricuspis occurred in parts of Florida. Researchers such as S. D. Porter and L. W. Morrison wrote reports on initial establishment and on how flies spread from release points. Programs in Texas added Pseudactyon curvatus and other species to match different fire ant populations. The effort did not aim to remove fire ants in one season. The goal was to add a natural enemy that would change ant behavior and reduce foraging pressure over the years. These early steps set the pattern for decades of careful releases and monitoring across multiple states. International partners continued to offer guidance, and funding supported fieldwork and laboratory studies. But while the field trials hinted at success, the real mystery unfolded in locked labs. How did scientists actually smuggle a parasite of fire ants into America without disaster? How the flies were brought here. When the first candidate fly species passed quarantine tests, the work moved into secure rearing rooms. Researchers from the United States Department of Agriculture Agricultural Research Service, the University of Texas at Austin Brackenridge Field Laboratory, and Texas A&M University set up programs to raise, transport, and release Pseudactyon flies. Partners in Argentina and Brazil continued to provide field guidance and source material for the fly species that matched local fire ant types. Rearing phorid flies is delicate work. Labs kept live fire ants to feed fly larvae and to supply workers for parasitism. Technicians learnt to spot parasitized ants by the small bulge that forms where the fly pupa develops. Some facilities used an automated attack box and humidity controls to keep conditions stable while flies completed their life cycle. Staff fed ant colonies sugar and protein and moved parasitized workers carefully to reduce accidental deaths. They used two main release methods. One method placed small cages of parasitized ants directly on active fire ant mounds. The cages let adult flies emerge near others so the flies could find hosts on their own. A second method released adults reared in the lab near foraging trails or mound edges. Transport rules were strict. Permits required that parasitized ants move under secure conditions between facilities and release points. Quarantine procedures used double doors, fine mesh screens, and careful handling to avoid accidental escapes. State agricultural staff and federal reviewers inspected facilities and approved transport plans before any field release. Extension educators and county staff informed landowners and local workers so that release sites were ready and monitored. Monitoring started as soon as releases began. Sticky cards and modified traps captured small adult flies as they flew near baits or mounds. Aspirators helped workers collect flies from vegetation and from traps. Direct observation of ant trails, with counts at bait stations, provided data on how foraging changed after release. Researchers checked traps daily during the initial weeks, and then on a regular schedule. Each capture and field observation entered a shared record that technical teams used to track establishment and spread. Pseudactyon tricuspis was among the first species released in North Florida between 1997 and 1999. Later programs added species such as Pseudactyon curvatus and others to match different fire ant forms and to expand coverage across states. In Texas and the Gulf Coast, researchers selected smaller fly species for polygyne fire ant colonies that produce many small workers. Matching fly species to the ant population proved important to improve the chances of establishment. Researchers adapted methods over time. When sticky traps proved effective, the standardized trap designs and sampling routines were used to compare results across counties. Release numbers and timing changed with early results. Where a pilot release did not produce many recaptures, laboratories adjusted rearing rates and release schedules. collaboration among the United States Department of Agriculture, Agricultural Research Service, the University of Texas at Austin Brackenridge Field Laboratory, and Texas A&M University helped spread technical knowledge and supported wider monitoring efforts. Private landowners, pest control operators, and university extension agents sometimes hosted release sites or provided access to fields. International cooperation remained part of the program as South American colleagues identified new candidate Pseudactyon species. The combined effort over decades created the careful, measured release program that moved the flies from quarantine rooms into the landscapes where fire ants lived. Records and maps showed each release location with GPS coordinates. Seasonal timing mattered because flies were most active in warm months. Field crews coordinated with landowners to keep records and to protect monitored mounds during baiting events. Over the years, these small steps added up into detailed data sets that guided future releases and refined methods over time. But after all these years of careful releases and endless data collection, the real question remained: would these tiny flies actually change the fire ants' world in a measurable way? What happened to ant behavior? Scientists measured how phorid flies changed the everyday life of red imported fire ants. Field specialists watched foragers, set baits, and counted workers before and after fly releases. The first and most obvious effect was on foraging. When flies were present, fewer ants left the nest to find food. Workers who did leave moved in short bursts and often stayed close to cover. They avoided open trails and open ground where the tiny flies hunted. The change reduced the amount of food carried back to the colony for minutes, hours, or longer during repeated attacks. A single fly attack often causes a large reaction among workers. Adult flies hover and then quickly land on a chosen worker. That worker will stop moving, drop what it carries, or run back toward the mound. In many field tests, a small number of fly strikes led to large drops in bait removal at feeding stations. In plots where scientists released Pseudactyon flies repeatedly, measured bait uptake fell compared with control plots that had no flies. The effect came more from behavior than from direct deaths. Lab work and field tests showed that a few deaths could trigger a wide fear response in foragers. When workers feared attack, they did less outside work, and the colony collected less food overall. Researchers recorded parasitism rates with advanced methods. They collected workers from foraging lines and from mounds and examined them for fly pupae. Parasitism rates varied by place and by season. In some sites, parasitism rose during warm months when flies were active. In other places, rates stayed low because the conditions reduced fly numbers. Even when measured, worker deaths were modest. The repeated presence of flies kept overall foraging lower over time. That constant pressure mattered more than occasional high mortality. The social form of the fire ant colony influenced the impact. Monogyne colonies have a single queen and different worker patterns from polygyne colonies that contain many queens. Polygyne colonies often produce many small workers and can replace losses faster. Where polygyne colonies dominate, some phorid species showed smaller effects on colony success. Researchers matched Pseudactyon species to local ant types to improve the match and impact. Choosing a fly that preferred the local worker size and behavior raised the chance of noticeable change. Ants also adjusted the time and place of their activity. Workers shifted foraging to shaded paths under vegetation or to times of day when flies were less active. Some colonies increased night foraging to avoid daytime attacks. Those shifts let colonies keep gathering some food, but at a lower overall rate. Scientists mapped worker routes and set bait stations at different distances and covers. The maps showed clear route changes and different bait removal patterns between sites with and without flies. The change in ant behavior altered local competition. In plots with persistent fly activity, native ant species sometimes increased their presence at baits and along trails. With less fire ant activity, other species could take small food items and occupy micro-habitats once held by fire ants. Researchers recorded shifts in seed removal and in predation on small arthropods. Those effects depended on habitat and the mix of native species at each site. Some places showed an increase in native ants foraging, where phorid flies had been active for seasons. Long-term studies tracked colonies for months or years. In some cases, researchers found slower colony growth, lower brood production, or smaller worker size where flies were abundant over multiple seasons. In other cases, colonies showed little long-term decline because they adjusted their behavior or accessed food during low fly activity. Disease, weather, and other predators also affected colony change. So teams kept careful records to separate the effect of phorid flies from other causes. Experimental designs used paired plots and repeated measures to show cause and effect. Scientists compared similar plots with and without fly releases and measured bait removal, worker numbers at stations, and brood development. They used traps to catch adult flies and counted parasitized workers. Those paired studies produced consistent results about behavior: repeated fly attacks, lower foraging, and force behavior change. Whether those changes led to colony failure depended on many local factors recorded in the trials. Field reports by researchers such as S.D. Porter and L.W. Morrison provided concrete numbers in trials. In monitored plots, bait removal fell by measurable amounts during active fly periods, and native ants increased activity in plots. Other researchers reported seasonal peaks in parasitism and clear links between repeated fly pressure and slower brood growth. These empirical records helped build the long-term data sets that clearly show how behavior change develops over time. But numbers on a page only told part of the story. The real test came with time, because if flies could reshape behavior in weeks, what might decades reveal? What changed over decades? After the first releases, the program expanded along the Gulf Coast and into the Deep South. Scientists from the United States Department of Agriculture Agricultural Research Service, the University of Texas at Austin Brackenridge Field Laboratory, and Texas A&M University coordinated many releases. They worked with state agricultural offices and with university extension services. Scientists added more species of Pseudactyon to match different fire ant types. Early efforts concentrated in North Florida and East Texas. Today, established fly records include many counties in Florida, Texas, Louisiana, Alabama, Mississippi, Georgia, and South Carolina, where monitoring shows recurring captures and sustained presence in warm seasons. Researchers keep mapping the spread and planning new targeted releases. Field monitoring over the years showed clear patterns. In some counties, Pseudactyon tricuspis and Pseudactyon curvatus established breeding populations that spread outward from release points. Natural dispersal moved flies at rates that changed with weather and with the type of land cover. Flies spread faster in warm, humid areas where adults survive and reproduce well. Cold winters and dry spells limited numbers in northern and inland pockets. Those limits produced a patchwork of strong and weak zones across the southern landscape. Matching fly species to the local ant population proved important. Fire ant colonies differ by social form and by worker size. Polygyne colonies have many queens and many small workers. Monogyne colonies have one queen and larger workers. When researchers released a Pseudactyon species that preferred a particular worker size, the chance of establishment rose. Researchers used surveys to determine the dominant colony type and chose fly species accordingly. South American collaborators continued to supply candidate flies and field knowledge. Effects on other insects and on small food webs were noticeable in monitored plots. In areas with persistent fly activity, bait removal by fire ants fell, and native ant species often increased their activity at baits. Seed removal by native seed eaters rose in some plots where fire ant foraging declined. Predators such as ground beetles and small birds showed small differences in where they foraged in sites with many fly attacks. Those effects varied with habitat and with the local mix of insect species at each site. Long-term trials asked whether flies could lower fire ant numbers at the landscape scale. Results varied by place. A single Pseudactian species released at one location rarely removed fire ants across a whole county. Where scientists released several fly species over time, and where other natural enemies or local control measures were present, researchers saw larger and more persistent reductions in foraging pressure. In some sites, the combined pressure from multiple flies and occasional baiting produced clearer reductions in colony growth and brood production over several seasons. Methods improve with experience. Scientists standardized trap designs, sampling intervals, and data recording so that results from one state could be compared with those from another. Rearing techniques became more reliable, producing cohorts of adult flies timed for release during peak ant activity. Researchers began to use global positioning system coordinates for release mounds and to log weather conditions at the moment of release. These steps made it easier to track the spread and to evaluate which release strategies gave stronger establishment. Funding and partnerships sustained the work. Federal research funding supported core laboratories. State agencies and university extension programs provided field staff and local outreach. Private landowners and pest control firms sometimes hosted release sites or shared local observations. International collaboration with entomologists in Argentina and Brazil remained important for locating new Pseudactian species and for sharing field knowledge. The program depended on steady cooperation among many institutions and individuals. After 3 decades, the program left a measurable mark. First, Pseudactian flies are now established across millions of acres in parts of the southern United States. The presence of multiple fly species in a region gave the strongest behavioral pressure on fire ants. Second, where flies became common, ant foraging dropped and worker routes shifted. That change allowed native ants and some other insects to increase activity at certain baits and sites. At the same time, the record showed limits. Climate barriers left gaps in the map where flies could not persist. Matching of fly species to local ant social forms required ongoing surveys and occasional new releases. A single fly species alone rarely produced a wide-scale collapse of fire ant populations. Researchers, therefore, treated the flies as one tool among several for long-term management of the invasive ants. The many years of work produced a rich data set of releases, captures, and behavioral measures. That record helps managers choose which fly species to release and where to focus monitoring in the future. After decades of careful releases, tiny flies reshaped the battle against fire ants in ways no one expected. But did this long experiment really succeed? Or just shift the problem? Should scientists keep releasing natural enemies? Or are we tampering too much with ecosystems? And if it works here, what other invasive species might be next? Let us know your thoughts in the comments section below. And don't forget to like, share, and subscribe for more interesting videos.